Welcome back to the deep dive. Today we're cracking open some really interesting source material you sent us.
Yeah, this is good stuff. It's transcripts and notes from a statistical genetics course.
That's right, the kind taught by Professor Guilherme da Silva Pereira at the Federal University of Viçosa Department of Agronomy. Pretty detailed.
And we're diving into, well, a really central topic through plant breeding today, genome-wide association studies. You know, GWS.
GWS, right, it's the super powerful tool for finding genes.
It is, but it works.
Yeah.
Well, it works quite differently than maybe the older methods you might know.
Exactly. So our goal here is to unpack these notes, really get a feel for how GWS lets researchers find important genes, especially in these really diverse groups of plants.
And how it stacks up against traditional linkage mapping.
Okay, so let's start there. Traditional mapping, that's usually with controlled crosses, isn't it? Like F2 populations or RILs.
Precisely. You set up specific crosses, you create these mapping populations, the relationships are known, the structure is clear.
But the downside is narrow genetics, limited alleles.
Very limited. Basically, you're working with whatever variation existed between the two parents you crossed. Good for finding major genes sometimes, but you might miss a lot.
Right. It's like looking for something inside one specific, very well-documented family tree. You know who's who.
But what if the trait or the allele you need just isn't in that family?
Ah, okay, so that's where GWS comes in, using these diversity panels.
Exactly. Think of a diversity panel as a big collection. Lots of different individuals can be breeding lines, land races, even wild relatives sometimes.
Yeah.
Much, much broader genetic base.
So potentially way more alleles for whatever trait you're interested in. It sounds like a massive kind of messy family reunion compared to the single family tree.
That's a good analogy. Messy is key. Because unlike the controlled cross, you don't initially know all the relationships, their structure in there, history, but it's not predefined.
And dealing with that messiness is where the statistics get, well, pretty critical.
Absolutely the fundamental. And one core concept you absolutely have to grasp is linkage disequilibrium, LD.
LDD, okay. And this isn't just about genes being physically linked on a chromosome, is it?
Not solely, no. It's the non-random association between alleles at different spots in the genome. So if you see allele A at one locus and allele B at another appearing together more often than you'd expect just by chance,
They're in LD.
they're in LD. And this association, it breaks down over time.
Because of recombination, right. Shuffling the deck each generation.
Exactly. The more generations of recombination you have in the history of a population, the more that LD decays. The blocks of associated alleles get smaller.
So in these diversity panels, with potentially long histories of mixing,
Much smaller LD blocks than in, say, a recent F2 cross where there hasn't been much time for recombination to break things up.
okay, and this is interesting because smaller LD blocks mean higher mapping resolution. You can pinpoint the gene's location more precisely.
You potentially can, yes.
Mhm.
Much finer mapping. But
Yeah.
there's a catch.
Always a catch.
To actually detect associations within those small blocks, you need vastly more markers scattered across the genome. We're talking millions, often.
Millions of SNPs, those single DNA letter changes.
Right. You need that density to capture the signal. And we use measures like R squared or D prime, to quantify how strong that LD is.
Got it. High resolution potential, but needs tons of markers.
Okay, but we still have that messy reunion problem, the unknown relationships in the diversity panel, population structure.
Yes, the next big hurdle. Population structure basically means your panel isn't one homogeneous group. It has subgroups, maybe based on geographic origin or breeding history.
And these subgroups might just have different frequencies of certain genes or alleles by chance.
Exactly. And if one subgroup happens to be enriched for a particular trait and also happens to have a high frequency of a certain marker, well, that marker might look like it's associated with the trait.
Even if it's not actually linked to the causal gene,
Yeah.
just guilt by association because they're both common in that subgroup. Precisely. That's a spurious association, a false positive driven purely by the underlying structure. Big problem.
Okay, so how do you fix that? How do you untangle the true signal from this structural noise?
You actually use the genetic markers themselves, the same SNPs, to estimate that structure first. There are methods like principal component analysis, PCAs, very common and fast.
So you use the markers to figure out the hidden subgroups.
And then you control for that structure in your statistical model when you test each marker for association with the trait.
Which brings us to the models. These aren't simple tests, I guess.
No, not usually. The standard approach now involves linear mixed effect models. Mixed models are powerful because they let you account for different sources of variation simultaneously.
Including the fixed effect of the marker you're testing and this population structure stuff.
Right. So typically you see what's called the Q plus K model, the effect of the individual SNP marker you're testing. That's treated as a fixed effect. You want to estimate its specific impact.
Okay, fixed effect for the S&P. What's the Q and K?
Q represents that major population structure we just talked about, the stuff you estimated with PCA perhaps. That often goes in as a fixed effect too, or as covariates. It accounts for those big subgroup differences.
Right, handles the major ancestry chunks. And K.
K is the kinship matrix. It's a measure of the genetic relatedness between all pairs of individuals in your panel, calculated across the whole genome. And this goes into the model as a random effect.
Okay, why random?
Because you're not interested in the specific effect of each pair's relationship. You want to account for the overall variance caused by the fact that related individuals will tend to have similar trait values just due to their shared background genetics,
Yeah.
not necessarily because of the one SNP you're testing right now.
Ah, so K accounts for that background genetic similarity across the whole genome, stopping it from inflating the signal of the single marker you're focused on.
That's the idea. It soaks up that background relatedness, that sort of polygenic resemblance, letting the fixed effect of the SNP stand out more clearly if it's real. Q handles the big structure. K handles the finer genome-wide relatedness.
Makes sense. It cleans up the picture,
No,
and
but
the results often shown in those Manhattan plots.
Yep, the classic Manhattan plot. Each dot is a marker. Its position on the x-axis is its genomic location, and the y-axis is the strength of association, usually like a death log 10 p-value. Significant hits pop up like skyscrapers.
But with millions of tests, you have to be careful about false positives, right?
Absolutely crucial. Multiple testing correction is non-negotiable. Things like Bonferroni correction or maybe false discovery rate, FDR control, you have to adjust your significance threshold.
Okay, so let's recap quickly. Traditional linkage mapping, known structure, limited diversity, maybe higher power for alleles in the cross, but lower resolution.
Right. And GWS uses diverse panels, massive diversity, potentially higher resolution thanks to historical recombination shrinking LD blocks.
But you have to deal with population structure and kinship using sophisticated models like Q plus K, and you need tons of markers.
That's the essence of it.
So GWS really is a powerful tool in the plant breeder's kit. It opens the door to finding useful genes within that vast natural diversity out there.
It does, by statistically navigating these complexities of recombination history and population structure.
Which really makes you wonder, doesn't it, how much incredibly valuable genetic variation is still kind of hidden away in diverse plant populations, stuff that maybe traditional methods just couldn't easily find.
That's the big question. And how can we keep refining these approaches, these statistical tools, to unlock even more of that potential for improving our crops? Something to think about.
Definitely food for thought. Well, that wraps up this deep dive based on those course notes. Thanks for tuning in.